So the bottom of the falciform ligament is called the round ligament, which is the same thing as the ligamentary teres.
Teres?
Teres, T-E-R-E-S.
Okay.
Teres. Which is my understanding of Latin simply means round. So it's the round ligament. On the right border, you're going to see a little indentation there, and that's where the gallbladder sits. And it's mostly underneath the liver. I'm going to put that as a dotted line there. And that's the cystic duct. So you see the gallbladder poking out here, but it goes back underneath, and it becomes a cystic duct out here. And I put that diagram on the board last time, so I'm not going to do all that again. Underneath the liver on the back side, you've got a lobe that's shaped a little bit like this, kind of curves like that in the back, and that's called the caudate lobe. Like I said, that's in the posterior. So you can't really see it from the front like that. And then underneath here, and I believe it's in this region, underneath there, you have a fourth lobe, and that's called the quadrate lobe.
Do we have to write the word necrotic? Just quadrate.
Quadrate lobe?
Yeah. Like on the
Whoa,
word
whoa.
thing, you have it as quadrant necrotic lobe. Do we have to have
You
the
mean-
word necrotic?
Oh, hepatic. No, I don't care. Hepatic just means liver. Yeah. So, no, these are all hepatic lobes. These are structures that you can see on the outside of the liver. Now, it's interesting, they don't follow the segmentation pattern inside the liver. Like when we talk about the lungs and the kidneys, you'll see that the outside lobes that we can see are mimicked with the vasculature structure inside the lobes. But in the liver, that's not the case. So, for instance, surgically, you can't dissect the left lobe away from the right lobe. It'll bleed, it'll hemorrhage. So I think there are 7 or 8 internal lobes inside the liver. I don't have them memorized. It's something that a surgeon would be worried about. Whereas in the lungs, we'll see the lungs, they either have 2 or 3 lobes, and they actually do have an air supply and a blood supply that goes to each individual lung. So you could actually separate those out and surgically remove them, but not in the liver. You have to follow a different pattern. Okay. So, anybody crudely aware of what the liver does?
Filter,
It's a filter. Yeah. Okay. What's a filter?
blood.
Blood. It filters blood. I think I mentioned something that it does. It does nutrient interconversion. Okay. And it does that because raw nutrient-rich blood coming from your digestive tract comes into the liver. So I'm going to erase some of this stuff here. I'll just put GI tract over here. The intestine, stomach, spleen, all of that. And we know that that blood comes in underneath. Oh, I probably should have done that. Change that to blue. And that's the hepatic portal vein.
Blood.
So that brings nutrient-rich but oxygen-poor blood into the liver. Okay. Nutrient-rich but oxygen-poor blood into the liver. The liver is a really metabolically active organ, so it needs a lot of oxygen. It's not going to get that oxygen from the portal vein. Instead, it's going to get it from the hepatic artery, which we know comes off of the celiac trunk. Off of the abdominal aorta. I got the common hepatic and then hepatic artery. Okay. And then lastly, coming out the back side of the liver, hepatic things. So, we get blood coming in two different places, and we get it leaving in one. And then within the body of the liver, some interesting stuff happens. If I take a chunk out of the liver, okay, if I just took a cross-section through the liver, an histology slice, I would see vertical columns of tissue arranged in the liver. And we call these lobules, hepatic lobules. And they're roughly hexagonal in shape. These really I try to draw in there in the green. That's something we went over last time. I got the gallbladder, it's connected to something, then it goes into another tube which runs down here. What do you suppose that is? Bile duct, right. This is our biliary circulation. So up there we've got the left and right hepatic ducts, common hepatic duct, cystic duct to the gallbladder, and then where those join, that's the common bile duct all the way down to the papilla and out into the duodenum. We went over that last time. Okay. All right. So I've got three vessels coming in the inferior surface of the liver there. So we call that the hepatic triad. Okay. So the triad consists of these three things. Bile duct, hepatic portal vein, and hepatic artery. That's your triad. And the vasculature inside the liver is maintained in that triad all the way down to this level. So what we do is we have finer and finer and finer branches within here. I don't know how many generations of branching. I would guess it's probably a dozen or more, okay, that are branches, until we get down to this level. So here we're looking at histology. Okay. So this would be, say, 100x histology. We've magnified it. We're looking at a lobule. The lobule is essentially the functional filter of the liver. Okay? And it's made mostly out of hepatocytes. I'm not going to draw them all in, or my picture will be so busy I won't be able to see anything in it. I'm going to put a couple rows in here. Okay, so we've got these hepatocytes, liver cells. You've got a picture of this in your book. You got your lab manual open?
Yeah.
What page is that on there?
504.
505?
4.
504? Okay.
Well, there's one.
If you want a picture that's way better than my drawing, take a look at that. Well, I like to do this because I'm building it. I'm putting it together instead of just showing you a picture. So here's your lobule. Those cells are called hepatocytes, liver cells. So the hepatocytes are the functional tissue of the liver. They're what do all the liver stuff. Okay? They synthesize all those blood proteins, for instance. Okay? They synthesize albumin. It's one of the main things that the liver does, is synthesize albumin. They make clotting factors. Okay? They make fibrinogen. All that stuff is synthesized by hepatocytes. They also take in the raw nutrients and convert it to things that we can use. So they do nutrient processing too. Another type of nutrient processing is identifying waste. So if they bring in things that are toxic, the hepatocytes recognize that, tag it with a material called ubiquitin, and then secrete it back into the blood so the kidneys know to get rid of it. Okay, so the The liver recognizes toxins and then tries to conjugate it to a molecule that the kidneys will recognize as waste. Basically, put a label on it that says get rid of this. Okay. Well, at the corner of each one of these hexagons, we have a branch of the hepatic artery. That's an arterial. And we should have a branch of the portal vein. And that's usually mostly collapsed. Call that portal venule. And then lastly, we should have bile ductule in there. And the bile ductule tends to have cuboidal cells that make up its wall rather than simple squamous epithelium. And these aren't just floating in space, they're usually in some elastic tissue and some connective tissue. So let's typically see some connective tissue around this. Kind of holds it in place like that. And that filament of connective tissue may run along here. And then ideally you get another triad over there, and the same thing here. You get the idea. Okay. You get a triad at each one of those vertices. In theory. Okay. In reality, it doesn't work out quite that nicely. Okay. But that's what we hope to see. So blood is coming in, guys. Blood is coming in the hepatic artery and in the portal vein. And now it does something that, to my knowledge, this is the only place in the body blood does this. They mix. Okay? The vein and the artery mix. And they flow down between the rows of hepatocytes in what's called a sinus. Okay? Remember we learned about the types of the capillaries? We had pardon me. We had continuous, then we had fenestrated, and we had the sinusoidal ones. And the sinusoidal ones were the real leaky ones. This is where we have sinusoidal capillaries, and that's what these open spaces are here. The sinusoidal capillaries are lined by columns of hepatocytes. The columns of hepatocytes are called cords. So hepatocytes in cords. Clear enough on that? What a cord is? It's just a row of hepatocytes. Well, the blood flows into the middle of each one of these lobules, and that's what I've tried to put for you here. And then this becomes the hepatic vein. The actual structure in there is called the central canal. But it's eventually going to become the hepatic vein. If you jump back over here for a minute, that's where the blood exits the liver, goes into the, if I put IVC here, you know what I'm talking about? Inferior vena cava. So that's where we're going to go back into the heart. So let's pause for a second and think conceptually. We took a lot of nutrient-rich blood from our digestive organs. We brought it into the liver, disseminated it to the lobules, passed it through the sinuses, where now the hepatocytes absorb the raw nutrients, process it, put it back into the sinusoidal capillaries again, and so then what arrives here is processed nutrients. And now that goes through the hepatic veins into the inferior vena cava, and now we're ready to spread that blood around the body. Okay. That makes sense? Of course, I'll lecture on this a little bit in lecture too, but this is just to introduce the idea. So you're going to be looking at all these parts today. I want you to have some idea what you're looking at. What's something else that hepatocytes do? So they do detoxification, nutrient interconversion, they do blood protein synthesis. What's a fourth thing that they do? What's something that the liver produces? Well, that's one of the proteins. Bile. The hepatocytes are actually making that cholesterol salt called bile. Well, bile we don't want to put into the bloodstream. Bile we want to put down